So, good morning, everybody. It's my pleasure to welcome you here in the auditorium and live on your TVs, on your laptops, on your phones. Just a quick reminder that we do have the Q&A system through the app or the website. And as you've seen yesterday in the keynote, in the Taylor Ruck lecture, there's this upvote feature. You can post questions. And I hope that this will also allow some of the virtual participants to interact with our speaker, who is now going to be introduced by my co-chair, Thomas Yeo.
Okay, it is my pleasure to introduce our first keynote speaker, Heisenberg professor Sarah Genon. Sarah is a group leader of the Cognitive Neuroinformatics Lab at the Institute of Neuroscience and Medicine at the Research Center Jülich in Germany. Over the past 10 years, Sarah has made important contributions to understanding of brain organization and brain behavior relationships. So for example, I was really intrigued by her 2019 eLife study, showing that the associations between a wide range of psychological tests and structural MRI have small effect sizes. So this in fact suggests the need for much larger sample sizes, which foreshadows the recent provocative BWA study. So today, Sarah is going to tell us more about the principles and challenges in mapping brain organization and brain behavior relationship. So without further ado, here's Professor Sarah Genon.
Thank you very much, Thomas, for the nice introduction, and thank you very much to the organizer for having invited me. I'm really happy to present our work today. So before jumping into the challenges in brain behavior mapping, I would like to first address the complexity of understanding brain organization. This can be well illustrated by one of the most studied regions in neuroscience, which is the hippocampus. So it receives a lot of interest in our community because it shows alteration in the most frequent neurocognitive disorders, such as Alzheimer's disease, depression, schizophrenia, but also from a basic neuroscience standpoint. From a phylogeny standpoint, it may hold some keys of the principle of brain organization as a relatively old region. If we look at parcellation and gradients map of the hippocampal formation that we can find in the literature, we see very complex patterns. We see a lot of heterogeneity. And to try to better understand this heterogeneity, here I organize the parcellation and gradient based on the type of features the study use. And on the left, this is studies which use connectivity features. And on the right, these are studies that probe microstructural features, local features. And the basic idea to understand the complexity of the heterogeneity and the heterogeneity of the patterns here is that if we consider features that probe into local microstructure, such as cytoarchitecture, which typically probe properties of local processing of information, we see typically this organization in the hippocampus along the medial lateral axis. It's like one organizational dimension. Then if we look at connectivity markers, we see this anterior-posterior differentiation. All right, so now with this in mind, now this can be illustrated with meta-analytic connectivity modeling, this anterior-posterior differentiation and resting state functional connectivity. In particular, we really see this, at three-cluster solution, we really see this head, body, and tail differentiation, which is well known in the hippocampus. But then we may think, well, this is because we use MRI, so it's a completely different technique than histology. But we also look at structural covariance based on gray matter volume from structural MRI, which is known to probe functional coupling between brain regions. And we show an anterior-posterior differentiation as well. But already at a three-cluster split, we see this medial lateral differentiation in the posterior part of the hippocampus, which really resemble the differentiation which is known between CA and the subiculum from cytoarchitecture. So this illustrates how to look at this variety of parcellation and gradient study. Basically, the extent to which the features step into large-scale functional integration versus microstructural properties will determine the extent to which either the anterior-posterior differentiation or the medial lateral differentiation would be expressed. And in between the two extremes, we have structural covariance as a relatively hybrid technique. The question now could be, why does it matter? Why we could not just focus on the hippocampal subfield model? It has been very useful in many applications. However, I think that to better understand the pathophysiology of some neurocognitive disorders and the behavioral phenotype that we see in these neurocognitive disorders, we need to consider all organizational dimensions as they are superimposed in the brain and here in the hippocampus. So to illustrate that point, I will show you a recent study that we did. So here we parcellate the hippocampal formation based on metabolic network from PET data. And so we had this anterior-posterior differentiation and medial lateral differentiation, so both emerge from the data. And each region has its specific metanetic network. Here we have illustrated the behavioral aspect associated with 3 networks based on activation database. And we can see that the red region, which more or less corresponds to the anterior subiculum, is associated with behavioral aspect pertaining to self-centric processing, while the yellow region here is associated with processing of more internal information like emotion and pain. And the blue region, which more or less corresponds to the posterior subiculum, has this very wide cortical network, which is more action-oriented way of processing or allocentric way of processing. Then we also look at how these metabolic network relate to neurotransmitter networks to have an idea of how this could relate to pathophysiology and possible action and treatment. And here we see, for example, that anterior subiculum, so the red region versus the blue region, are associated with different neurotransmitter profile. They are targeted by different neurotransmitter system. As another illustration of how this anterior-posterior differentiation may be important for understanding behavioral phenotype, here what we did is that we took hippocampal seed region from structural covariance-based parcellation, and we use a data-driven approach, a multivariate mapping between this seed region, voxel in the brain in gray matter, and we include this in this multivariate approach behavioral symptoms. So this was done in a cohort of almost 400 patients with depression. And so this is data-driven, this is multivariate, and we have a cross-validation scheme. This is important for what I'm going to talk about later. And what we see here is that this approach really identifies a latent dimension which put on one pole the anterior hippocampal seed, and together with symptoms such as anxiety, and on the other pole, posterior seed with behavioral aspects such as insight. So this really illustrates how this anterior-posterior differentiation, organizational dimension may be important for understanding different behavioral phenotype, including within a single neurocognitive disorders, such as depression here. Now, from there, I will move more into the question about how we, in general, relate brain to behavior, in particular in a healthy population. So in the last decades, the advent of neuroimaging scanner and the computation, the possibility of computing different structural features from MRI data, in particular gray matter volume and cortical thickness, opened a promising perspective for relating inter-individual variability in behavior to inter-individual variability in brain structure. And this can be seen from this influential piece from Canay and Rees a decade ago. Accordingly, we see in the literature many different associations. For example, the number of Facebook friends have been related to local gray matter volume, as well as political orientation has also been related to local gray matter volume in LC participants. And local corticness also show association with aspects such as theory of mind or music pitch performance. So these are just a few examples of the many associations that you can find in the literature. However, some years ago, we started having some anecdotal evidence of possible replication issues in brain structure behavior association. So, for example, here we find an association between performance, visuospatial performance at the TMTA and gray matter volume in the dorsal premotor cortex. And it was in what at that time we consider as a relatively well-powered sample. That was, the association was significant, and from a neurobiological standpoint, it makes sense, and this was well aligned with the human brain mapping literature. However, when we try to replicate this association is in an independent but demographically similar sample, we could not replicate it, and it was not because it was not significant, because the correlation coefficient was basically null. So from there, we started a more extensive evaluation of the replicability of association between brain structure with local gray matter volume and behavioral association. Basically, we did resampling within cohort, and when we found a significant association, we looked how much time across split we could replicate this association. For the replication, we used different indices. One of them was the direction should be the same as in the discovery sample, and it should be significant. But we also used the base factor. We had a relatively large cohort of more than 400 LC subjects, so that was the NKI data. And for a measure that show a significant association initially in that study and in many studies such as perceptual IQ, what we found, first I need to say that blue here will represent replication success, and orange color will represent replication failure. And what we saw across these 100 split is basically replication, majoritarily replication failures. And this was the same when we used the base factor as a replication index. We mainly find evidence for the null hypothesis, so for the hypothesis of no association. And for the sake of comparison, so this replication issue is particularly in healthy population, because if we look at a clinical sample in which conceptually the idea might be a bit different. Here we're mapping behavioral deficits to gray matter atrophy. And there in a clinical sample for a verbal learning performance task, we found way better replication rate for both indices. So we are way more on the blue side. From there, we started discussing a possible replication crisis in the field. However, we got several comments and concerns that this really could be premature because this might be related to the sample that we use and to the fact that we use local gray matter volume, which could be considered as a relatively crude method. So to address this concern, we also look at cortical thickness in the HCP data, which is known as a high-quality data, and which also include composite score, which from a psychometric standpoint could be considered as showing more, possibly more validity. I will show you another approach that we use, which is looking at spatial consistency across splits now. And as a benchmark here, we use age. So you can see that for age, we have a relatively high rate of spatial consistency across the different replication attempts in the brain, so in particular in the prefrontal cortex. However, for measures such as delay discounting task and conscientiousness, we see that the replication rate in terms of spatial consistency are very low. And we had a similar pattern as well with gray matter volume in the previous study. I just didn't report the result here. And since that, and also since there are no other very important studies like the study from Marek and collaborators, we now do discuss a replication crisis in the field. We did that in a recent piece. But we also highlighted possible solutions, including the use of large data set. We discuss important issue related to confound removal. We promote the use of multivariate approach, the use of a cross-validation scheme, and very importantly, out-of-court replicability evidence. So with this guideline and aspect in mind, we look again at association, but now multivariate association between behavioral data, a range of behavioral data from HCP, and brain structural data, including cortical thickness, surface area, and gray matter volume. And we use, for the multivariate mapping, we use a regularized CCA with outer cross-validation and in inner cross-validation. This is a framework developed by Hagoston Mailik in UCL, code are in open access, so you can look at it and use it. But very importantly, we needed out of sample or out of court here replication data set. And so we needed a data set that has similar behavioral measurement than HCP young. And for that purpose, we used the HCP aging data set. And what we found is that we found one significant and very stable latent dimension in HCP young that on the behavioral side basically put on one pole cognitive control executive function and on the other pole negative behavioral aspects such as impulsivity. And we find a similar pattern in HCP aging. But here, this latent dimension mainly capture inter-individual variability in executive function or cognitive control. And when we look at the correlation of the behavioral loadings, they were significantly and highly correlated. So to some extent, we have a relatively good out-of-court replicability. At the brain side, these latent dimensions show association with higher region from the cortical hierarchy, but also sensory motor and more primary regions. So basically, it shows association with a dimension of differentiation along the cortical hierarchy, if you think of Daniel gradients, for example. And very importantly, the brain pattern were very similar across the two cohorts. And again, the loading were relatively well correlated. If you want to know more about that study, please visit poster 595 and just talk with Eliana Nicoles and Sobeski. She can really tell you more about her study. because now I will move to another topic, which is how we relate inter-individual variability in psychometric data to resting state data. And more particularly, how we predict inter-individual variability at a given psychometric variable, or we predict it based on inter-individual variability in resting state functional connectivity. So this line of research is important for basic neuroscience, but also more generally in the context of brain-based prediction, the deployment of brain-based prediction model for various applications in the society. And in that context, there are several ethical aspects that should be considered. So we need to have transparent model, which means that we need some interpretability from a neuroscience standpoint, which implies that the replicability of the brain-behavior association pattern is important. And another very important question is the generalizability of our models. So first, the question of interpretability is a very complex question in the multivariate context of the prediction. Some studies are hypothesis-driven, so they just focus on a specific set of region or network for the prediction. So for example, studies in psychiatry, they will just focus on the DMN or on the socio-affective networks. So there the question of the interpretability is relatively easy. But from a more data-driven approach, we would like to look at the whole brain and to identify or to better understand which region and connection play a role in the prediction or drive the prediction. To do that, some studies have taken what I would call a postdoc approach, in which they look at the weight assigned from the models to the features. However, in the multivariate context in which the features are correlated, the weight, this approach can be really misleading. The weight magnitude doesn't reflect the region association strength with the psychometric variable. So it is really hard to get neurobiological insight from these models. And one approach that we thought of, which is a relatively simple approach, but which also could be considered as a reductive approach, but it does actually work well. It's a region-based approach. So the idea is that if we take the vector of connectivity of this region, the premotor cortex, if we have the connectivity pattern across subject of this region, how well can we predict a cognitive variable? Here we have basically no predictive power in terms of prediction accuracy. But if we take another region such as the supramarginal gyrus and we try to predict this cognitive variable, now we have a way better prediction performance in terms of correlation between predicted and observed values. So with this approach, we could look at two perspectives. First, brain regions perspective, in which we can draw the psychometric profile for a given brain region. We did that, for example, for the primary visual cortex, Broca region, and the anterior hippocampus. But also from more psychometric variable perspective. So if we are interested in a specific psychometric variable, we can look across the brain, or prediction performance varies across brain regions. And we have done that for crystallized cognition and two different variables derived from the two backtasks in HCP. So first, for the first perspective, the psychometric profile, I will here focus only on Broca's region, because there we can already compare just the left Broca and the right Broca. So what you see on this radar plot, so first on the gray, this is the performance using the whole brain connectivity information, so the whole connectivity matrix. And the blue line is the performance for each behavioral variable when focusing on either the left or the right Broca connectivity. So of course, we don't reach the same performance as when using the whole brain because we don't have as much information as when using the whole brain. We have reduced the information. However, we see that for both left and right, we could to some extent predict reading and picture vocabulary performance. And in the left BRCA, we achieve similar performance than the whole brain model for performance at the language task, while in the right BRCA, we achieve relatively good performance at the two-back task. Now, if we stay with the two-back task and we look at the distribution of performance across brain region, we see, of course, the right BRCA, but also the supra region around the supramarginal gyrus. We also see the anterior insula and a region in the middle cingulate cortex. And this is for general accuracy at the two-back task. But we also look at the profile of prediction across region, specifically for performance for the face material. And we see similar region, which is good, but we also see in addition region in the fusiform gyrus and lateral temporal cortex. And these regions correspond to regions that are usually reported for face processing in the literature. So to some extent, it harked for the neurobiological validity of the model. And this is really important here for brain-based prediction. I, of course, only show the result where the pattern matched the human brain mapping literature, or makes sense from a neurobiological standpoint, but we also find cases in which the region that were predictive actually didn't really make sense, and then we look at how a model and we figure out that the model were driven by some confounds, and then we did confound removal, and then how a model was more neurobiologically meaningful or had more neurobiological validity, in the sense that we use the information that we think that we use. Okay, but the question then is also for these approaches, what is the replicability of the brain behavior pattern? And addressing this question is difficult because we need different data sets that have similar behavioral measure. And one only measure for which is possible is fluid cognition. Here we use 4 data sets, GSP, NKI, HCP aging, and HCP young. First, in GSP, unfortunately, the prediction performance were very low. But in the 3 other data sets, we see some similarities in the map of prediction. So we see, again, region around the supramarginal gyrus, in the cingulate cortex, and in the anterior insula. And these regions are known to be a crucial hub in the brain and also a region of high interindividual variability. So this suggests that we could focus on this region as a specific feature for sample who would have a smaller size, for example, for which we would need to reduce the number of features. But then this has the question of the generalizability of the model based on this region. And to address this question, we basically train in one data set and test in another data set. Overall, the prediction performance were relatively low, unfortunately, and what we see is that where it's get a bit higher is for data sets who have similar acquisition protocol or processing pipeline like the 2 HCP data sets. However, we also see that it's get a bit higher when we train the model in the NKI data, which has a whole adult lifespan, whole population, a more mixed population data set, and then we test in the HCP aging. And this leads me to another important question in the brain-based prediction field, which is the generalizability of our model to minority population. So we all work in the field with the same data set, which are open access, and that's really great, and they reflect the population from which they have been acquired. But it means that most of the time they are dominated by white European descendant population. And so the question we address is whether there might be bias against minority population for this model. And to look at that question, my colleague Yingwei Li looked at the HCP data, in which, again, we have a range of behavioral measurement, but we have also mainly white Americans reported ethnicity and a small proportion of African Americans. She also looked at the ABCD data, which has neuroimaging and a lot of behavioral measurement for thousands of kids or pre-adolescents between 9 and 11. It's also majority white Americans participant. And the idea here is that Yingwei trained the model on the whole population, then she compared the performance of the model trained specifically in white American versus, so she trained the model in the whole population and she compared the performance when the model was tested specifically in white American versus when it was tested specifically in matched African Americans, so matched to the white American test participant. And what we find is basically that we have poorer model performance, larger prediction error in that case for African Americans than for matched white Americans. This can be seen here in the HCP data for one variable, which is spatial orientation. So in the blue, the blue violin is African American, the green violin is white American, and in red this is the difference, which is significant, and in white this is the null model. And we had this significant difference in the prediction performance across many different variables in HCP, and we saw a similar pattern in ABCD data, with many variables showing significant differences. The question then that we look at is, okay, we have this bias for some specific population which are a minority in the data. What could be the potential consequences concretely of this bias? And to address this question, Yingwei looked at predicted versus the predicted minus observed behavioral scores in the two populations. And here this is what she found, for example, in the ABCD data. She found that there were more over-prediction in African Americans, so which are in blue again here, white American are in green. So there were more over-prediction for African American for variables such as rule breaking and aggressive behaviors. By over-prediction, I mean that the model predict the score higher than they truly are. And we also find a pattern for similar pattern for related variables such as social problems. So the potential consequences of this bias could be that we use such aspect for the diagnosis of mental disorders. So ultimately there is a risk with this bias in the model that African American will receive more false positive in the population. But then the next question is, how can we reduce this bias? There are a lot of strategies that have been suggested in the fairness of algorithm literature. Here we only try one relatively simple strategy in which we work on the training data set, and we train the model specifically in African American in one case. In another case, we train the model specifically in white American. but with the same sample size as the model trained specifically in African American. And in a third case, we trained the model in a mixed population, but half African American and half white Americans. And we look at the bias for the range of behavioral measure that we had. And what we see basically is that when the model is trained specifically in African American, we have less behavioral variable that show a bias against African Americans. However, we still see many behavioral variables for which there is a bias. And we thought of two potential factors that could explain this remaining bias. First, on the brain side, for decades, our preprocessing pipeline, the template that we use, the atlas that we use, they have been optimized to fit the majority population of the data set they were derived from. So basically, they've been optimized for the white European descendant population. So this could potentially contribute to the bias, but also on the behavioral side, the psychometric tool that we use, they may show relatively limited validity for minority population and different socio-cultural background. So to conclude from all our work, on the brain organization side, I hope I could show you today that different brain parcellation reflect the complexity of brain organization, in which we have different superimposed organizational dimensions, and that it's important to consider all these dimensions to better understand behavioral phenotype and pathophysiology in different clinical population, and also in the healthy population. Brain structural behavior association using traditional approach appear to show poor replicability. There is clearly a replication crisis. But multivariate brain structure behavior mapping reveals new, global, robust, and apparently cross-cohort replicable patterns. On the connectivity side, connectivity-based prediction of behavioral phenotype could benefit from frameworks promoting interpretability, which would be important for the deployment of this model in the society. Unfortunately, they show relatively limited cross-cohort replicability and generalizability at the moment, so this is really something we should work on. And very importantly, there is a fairness issue for minority groups in this model that we should work on in the future. So to conclude, we call for more diverse data set to address replicability, interpretability, and generalizability in brain behavior mapping and predictive model in the future. With that, I would like to thank all my really precious collaborators in ULISH and beyond. And thank you very much for your attention.
Thank you, Sarah, for this amazing talk. So Simon and I have been monitoring the Q&A on the phone. So we have a few minutes for some Q&A. So the first question we'll start with is, Sarah, is posterior hacking, predictive hacking, and cross-validation hacking are just as easy as p-hacking. What do you suggest as a way out?
Sorry, I will look at the question on the phone because I did not understand the first part of the which type of hacking?
Oh, no. I think you know what's p-hacking, right? But for cross-validation, you can also cheat by repeating, changing your algorithm or hyperparameters until you get a good result.
Yeah, that's right, and it can also be difficult to identify. And there is a interesting paper from Dustin Shiner's group on bioRxiv about how this predictive model can be corrupted. So this is really an interesting, I think, point of research. That this model could be corrupted as well, and we need to be really careful. And I think that's why we need this interpretability quality of the model, which may be a bit against the idea of multivariate mapping and data-driven approach, but on the other hand, this will really prevent us from corrupted model and also from possibly not neurobiologically valid model.
Okay, and the current second topmost question now is, are we misled by focusing too much on HCP? So everybody works on HCP, but it's a very narrow data set. Do we have a problem?
Yes, I would definitely agree with that. That's a great resource. That's really high quality data. The phenotyping is relatively extensive. Yeah, the MRI data are really good, but we overfitting it at the moment. So all results that we have in the literature are, in our field, are mainly based on HCP. So we are reinforcing our belief somehow, and it would be really good to have more diversity in open data sets.
Thanks, Sarah. Another question is, for your comparison for the different races for the prediction, how were socioeconomic factors that influence brain development, how they accounted for, given that they're well-known race, socioeconomic status confounds?
Yeah, that's a very good question. First, I would like to emphasize that we did not look at races. We look more at ethnicity in the sense that we use the information of the reported ethnicity by the participant, not some genetic ancestry. So we actually look at the question of ethnicity, having in mind that these are different socio-economical group of the population. And we try to, a different approach, we try to regress different confound that we could have in the data, such as for ABCD, we had family income, but that was not enough for controlling the bias. But that is true that these socio-cultural, socio-economical factor certainly play a very important role in these bias that we see in the model.
Thank you. Another highly voted question. Do brain behavior links actually make sense given all the shortcomings in the phenotypical data?
The phenotypical data in, I guess, the
cognitive and clinical.
Yeah, in the clinical sample or in the LC population is not precise. I think it's in the, maybe in general in the psychometric data. That for personally, yeah, I do think that the limited validity of psychometric tool could explain some part of the poor or relationship that we have, of the small effect size that we have, of the noise in the data, and of the replication issues. But I think there is also noise on the brain side. It's not only in the psychometric measurement. Our brain measurement might also not capture enough relevant signal for brain behavior association. I mean, signal in a statistical sense.
Right, kind of, there's a similar question on that, which is that most cognitive tests measure unnatural behavior. So how can we address brain behavior relationship with more natural behavior?
Yeah, in psychology, we call that more ecological measure. Yeah, this is really hard. I'm not aware of any data set in which this would have been more work on, so having more ecological measure, but that's an interesting idea, I think an interesting point to work on, to have more naturalistic measure, more ecological, psychological measure.
Well, thank you very much. We are at the end of the session. Thanks for the wonderful talk. Thanks for the questions that everybody asked online. Thanks for the upvoting. This was now seeing the system in real life, and it worked beautifully. And enjoy the rest of the conference. Thanks again, Sarah.
Thank you very much.